Did everybody hear that part?
Yeah.
Yes.
You're all under surveillance. Okay, cool. Let's do this. I'm gonna start right on over here. So remember what we did last time. And so I wanna make sure that I get my cool colors again. There it goes. One of the last sets, the first sets of classification is by us using a little chunk of DNA, right? This was our double-stranded DNA, right? And then for us, we would go into transcription and make out one little chunk of RNA, which was single-stranded RNA. So we call it transcript. And then through translation, right? This would turn into, whoops, lost my little pointer. Some sort of functional unit, you know, that we call protein. Now, that was what we call that positive direction of the central dogma. Everything proceeded pretty straightforward. I'm allowing some people into the room, so. All right, just needed to let some more people in there. Okay, so that meant a kind of positive direction from top to bottom, if you will, from a standard piece of DNA to a functional piece of protein. That's what's supposed to happen. Now, the negative sense, remember, we were talking about, in this case, using the complementary sequence, if you will, the one that did not give us an actual product. And so let me start writing that down. I'm going to use hopefully a slightly different color. I'm going to draw it off to the side. So now envision that this guy, instead of being positive sense, we're going to call it negative sense, and is the complementary version to that guy. I'm going to write it as comp, right? And so that one does not give us protein. You cannot create a full-blown protein out of that. So the only thing you can do is convert it into the positive sense. So what these particular types of viruses do is actually take that sequence, over here, and then convert it into positive sense single-stranded RNA as well. That happened over here.
Chris, if you can hear me, you're frozen.
Thumbs up, people, to make sure you guys can hear me.
Now we can.
Not sure what happened. All right, let's try this again. Having fun with my doodles, and apparently it got crashed. All right. Can everybody see my awesome doodles? Cool.
Yes.
Excellent. Thank you, guys. All right. So that was what we were talking about in terms of negative sense. And then we talked about a couple of variations in which you can go backwards into DNA, what we call reverse transcription, and so on and so forth. So be aware of those. So that was one way to classify. We use its genome, whether it's DNA or RNA or single-stranded or double-stranded in negative sense, positive sense, and so on and so forth. That's one version of that. Now, the other thing that we're going to start looking into is specifically what type of hosts can this organism infect. Now, understand that out of the cajillion viruses that are out there, okay, we can't really talk about every single host and every single combination. So we're just going to give you groups of them, and you are not required to memorize those. Understand that. Okay, you need to know that we use hosts as a way to classify them, not memorize the hosts. But I'm going to give you a few different examples. So how do they find the hosts? And this is something that has been popping up a lot especially due to the coronavirus stuff, right? And so, first of all, a virus can only actually try and kill you if it can get into a cell. Just because it's, somebody sneezes it on your face or licks you or whatever happens these days, doesn't mean you're going to get infected. For something to consider to be an infection, not only does it have to land on you, it has to attach itself on you, and then it has to survive your defenses. That's the only way that an infection really works, and we'll talk about that during the immunology and epidemiology lectures. So just because, again, somebody sneezes in your face doesn't mean you automatically get sick. Now, if it lands on your face or your hand or whatever it is, now that virus has to find its host cell. And the way that this happens is by this kind of what I like to call a handshake, in which one hand from the virus and the hand from the host kind of have to agree that they match. So this is your bacteriological and virological version of match.com, okay? You have to find somebody that's compatible and then agree, and then you can let them in. Wait, no, that's too much. Never mind, let's change that, whole different story. What has to happen is that there has to be a synchrony, a match between the virus and the host itself. In other words, to simplify that, if you would have something like, let's make this up, a dog virus for whatever reason, right? One of the reasons why it doesn't infect you, even though it is a virus and the dog could feasibly lick you, for example, is that that handshake is not compatible, meaning that the proteins that are present on that virus do not or are not compatible with the proteins that are present on you as a host. So not only does it have to land on you, but there has to be this interaction, which is what you're seeing on the lower right-hand side of that picture. The idea is that the host has these proteins on the surface of its membrane, and that the virus also has on its own. And so those two proteins have to interact. They have to kind of do that handshake. And if they agree, if they're compatible, then that virus can come in. That virion technically can enter. Now, sometimes it's one protein, sometimes it can be many, sometimes it can be 2 or 3. But that's what it's minimally required, is that there has to be this compatibility between the virus and the host for them to actually work out, okay? So that handshake, and this is what you're seeing over here. I think you guys can see my little arrow when I'm kind of drawing out. I'm gonna doodle it over here. And the interaction between the actual cell and the virus itself has to be a direct interaction between those proteins. If those again match, then the process of internalization occurs. Then the virus can, or the virion can actually get in. So these are your typical proteins that are doing so, okay? Now remember, virtually every single domain that we know, so all 3 domains, can be infected by viruses. All right, now again, you're not required to memorize these, but kind of wanted to give you a little bit of an example. I'll give you a couple of others later. For example, the group of viruses, the family called B.I.C, this case can infect both animals and plants. However, you'll notice that it doesn't infect something like bacteria, for example, right? And as you can kind of start scrolling down that list, you'll notice that these viruses can kind of jump across these domains, or sometimes stay within only one domain. Example, the Viridae, as far as we know, can hit all the eukaryotes. So it can hit you, plants, animals, insects, protists, so on and so forth, as far as we know. There are certain organisms that, as far as we understand, are incompatible, or these viruses cannot infect across the domains. But imagine if there was a virus that can jump across all of them. That would be a much bigger deal to truly worry about. All right. So what we're talking about in this case, again, is this handshake version that I'm talking about, in which the virion has some sort of protein on the outside, the host has some sort of protein on the outside, and then those two again can do this handshake. They can be compatible and interact, and allowing that virus to go in, which is the little animation that you're seeing on the right-hand side. Now, the third and final piece that we're going to address before we move on to cooler stuff is not only do we classify them by their genome, we classify them by what type of hosts they have, but we also classify them by their shapes. So viruses come in really cool shapes too. Remember that we're looking at first scale. Remember we talked about the size of these guys in comparison to everything else. Just reminders that there are certain viruses that are pretty large called the giant viruses, right? But the virus on the outside, the so-called virion that we were talking about before, can come in very unique shapes. And that is all due to the protein that they're composed out of, they're made out of in terms of the outside. And so that term, the coat that they're wearing, is referred to as a capsid. What does that mean? So we have typically 1, 2, maybe 3 different proteins that kind of like the Legos that we talked about before, just repeat over and over and over and over to make this enclosure, this casing. So you take a small little piece, let me make up a little square over there, that repeats multiple times. You guys can see my little doodles. And so now you take that guy and then you start assembling it. So what you're seeing, for example, in the very first version over here, let's see if we can get this guy going over here, right, is a virus that is referred to as a polyhedral virus. And what you're seeing is one little piece that is repeated over and over and over and over, hundreds, if not thousands of times, okay? And so we call that piece a capsomere. So that protein that's going to repeat itself multiple times is called a capsomere. And so when it assembles itself to give the shape of the ultimate virion, it's called a capsid. That guy right there. So what's the purpose? Well, like anything else, everything needs an enclosure. So it's pretty much a container, okay? But also it helps it attach to its host. So it has 2 main functions. One as a way to kind of contain it and hold it inside, package its DNA or RNA inside of it, but it also works in terms of recognition, that handshake that we were talking about a little bit earlier. And so the right-hand side, you're seeing a little bit of a pretty model done by computers on showing that there's one little piece. Let me see if we can circle it over here. There it goes. That repeats over and over and over hundreds, if not thousands of times, and turns into an enclosure. Now, what kind of shapes it can take, I'm just giving you a few little examples over here. You already saw the first one that we were talking about, the so-called polyhedral one. This can also be called a spherical simply because it's kind of roundish. But there are ones that look like little tubes. These are called helical. Okay. And there's obviously ones that we just can't declare shape, so those are usually referred to as complex. But probably my favorite one out of there are the ones that infect only bacteria, as far as we know. And these are called binal shapes. And they also are referred to as phage shapes. Whoops, went too far. The term phage usually is implying that it's going to infect bacteria, just so you know. And this is the one that you're seeing over here on the far right-hand side, on the top right-hand corner. Can't find a little marker over here. This guy or that guy, whichever one. And it kind of looks like a cool little lunar lander, in case you haven't seen that. And so guess where we got the cool ideas, right? And so in this case, it's a really cool system in which at the top, where the capsomere is, what looks like a little polyhedron, is where the material is stored. But the rest of that body acts like a little syringe, and it'll attach itself to the surface of bacteria and then inject its DNA into the actual host, this bacterium, and then take over the machinery. All right. So aside from the protein, some of these cells, including our awesome little current going on coronavirus, has something called an envelope. Now, not all of them have it, okay? So this is kind of, optional. So I'm gonna write it over here. And it can occur sometimes. And what we're seeing here is that not only the viruses will have a protein enclosure, a capsid, like we were talking about before, but they can also have an envelope, their own version of a membrane. Now, they don't typically make it. This is typically stolen. So when they're done killing their host or taking over their machinery, they kind of rip off a little piece of the skin of the host and take it with them. So kind of a messed up system. So usually when we're talking about these, we can call the viruses enveloped or non-enveloped, or if you just want to be cool and sound weirdo, or like a little weirdo, we call them naked. So keep that in mind. Now, what's the whole point behind ripping a little piece of skin? Well, probably the most genius side of this concept is the concept of a wolf in sheep's clothing. Is that if you can put on the skin of your enemy, you can kind of hide for a while. And so some of the viruses will, as they explode and take over the system, take a little skin around them and they'll hide from the host's defenses because they look like the host. They're taking their skin. So very much like a good old Texas Chainsaw Massacre kind of thing, right? So currently our version of SARS-CoV-2, the one that everybody's worried about right now, has that. It actually has a little skin on the outside, a little envelope with some proteins on the outside too. And we'll talk about that a little bit later too.
So I have a question.
Yeah, of course.
So the is the envelope the skin of the host, or is the envelope part of the virus?
Both, actually. So now, again, the virus doesn't normally come with it. It takes it from the host. And so as it exits the host, as it kills it, right, it'll take a little bit of it and throw it like a little coat on top of itself. And then it'll continue to use it until it finds a new host.
Okay, so it's separate from the capsid.
That is correct. It's outside the capsid. The virion itself is normally its genome and its capsid on the outside, and that's it. However, some of them can steal this extra layer of skin to kind of hide from the host defenses.
So can the same virus take skins from different things?
Well, keep in mind that anytime a virus is infecting, it's only infecting one host.
Oh, yeah.
Right? So it would only be able to take that skin from that particular host per se, right? But if that host, for whatever reason, jumps from one organism to another organism, this is something we call spillover. We'll talk about that later. Yeah, technically it could acquire different little, sheep's clothing. Cool questions, people. Cool questions.
Is this why people are asymptomatic with the coronavirus for like up to 2 weeks?
It could be. And so I'm not going to really establish that because I don't know enough about the virus since I haven't studied it too. But keep this in mind that while it's avoiding the host defenses, yeah, you're not going to feel a lot of the symptoms. It's also possible that you're not as susceptible to the virus, so it could infect you. It could be trying to take over you, but since it's not doing a good enough job, you're probably not going to feel very much of it either. Okay, so it could be a combination of these as well. And the other version of why it can take so long, 4 or 5 days, it could be because of the fact that it's still about to take over. It's still trying to find the cell to, infect. It hasn't been able to successfully do so. So part of that includes that time too. All right. So here's a couple of different orders that I just wanted to give you examples for. So again, mind you, I'm not asking you to memorize these, so don't try and do so. But so you'll notice that the first order that you're seeing there, which is Legamenna viralis, in this case, infects archaea. That's its hosts. So it's highly unlikely that it'll, spill over onto us. But, we've heard some worse things. And then as you can see, some of them are the other variations. And then you can also see the differences between the, whether they're positive sense or negative sense. But allow me to give you the order that some of you are going to be hearing a lot, which is 1, 2, 3, 4, 5, 6 of them from the top, which is the Nidovirales. That's the group that includes the coronaviruses, the CoVs per se. So these are positive strand, single stranded RNA viruses. So that way like I said, some of you guys have asked me to give a little bit of a talk. And so I've already constructed some of this stuff in there. So you guys can, if you want to join for that, you're more than welcome to listen to that story. All right. Oh, this didn't move. So let me give you a couple of differences between the DNA viruses, for example, and the RNA viruses, which are the next screen. So we have 2 groups of them in there, in which we have the double-stranded DNA versions, and then the single-stranded DNA ones. And even there's one that is kind of partial single and double. And you can see kind of what is included in there. So anything from your herpes viruses are double-stranded DNAs, viruses, sorry. And then here's a group of RNA viruses in there. You'll notice that a lot of them are single positive sense, although there's the bottom half are single negative sense behind that. So you'll see where these kind of go behind that, right? All right, so now we have three things that we want to look at in terms of classifying them: what type of DNA or RNA they contain, the host that they can infect, and then the protein on the outside. Those are the three main factors. Now, what we want to look at briefly, because again, we're trying to cover everything, is that we want to see how they reproduce. What is their life cycle? So in the past, we were looking at things like protists and worms and how they're actually going through the life cycle of entering the host and crawling through the skin, entering the lungs or something like that, and then laying eggs in and out of you and having fun. We have to look at their life cycle as well. And so as opposed to your classic life cycle that involves a host, well, viruses involve the host too, but they do it slightly different. What you're seeing here are examples of what we looked at in terms of our protists and our shrooms. Ours is kind of different because our viruses have two big stages. One of them on the outside, the virion stage, the one that is complete, right? And then the internal version of it, which is the virus stage. Now that means at some point in time, these virions or viruses, depending on which stage we're talking about, is being transmitted across intermediate hosts. And then eventually through somebody's wonderful not covering their face kind of thing, sneezes on you or whatever it is. And then once it makes it to you as the host, as the new host per se, then it has this internal stage of looking for that handshake and trying to get in. And hopefully there's this compatibility that can occur there, and then it takes over you. That's part of that fun. All right, so let's take a look at these guys, right? So there's 5 main stages, which are kind of self-explanatory, so it shouldn't be too difficult to kind of go along with this, okay? So first, it has to attach itself, pretty basic, right? Once it attaches itself, it has to gain entry into that host cell too. Once it gets inside, then it needs to start producing its own stuff, its own parts, its own pieces. So that's part of the synthesis stage. And then once it makes those, then it needs to assemble itself into its final shape. And then ultimately it still has to get out. And keep in mind that every single one of these stages is critical, because if one of these fails, obviously the virus will never make more of itself and never take over you or any other host. All right, so first let's look at the most common or standard version of this in terms of how a virus takes over and kills, right? So since we already have the 5 stages over there already listed, remember that that attachment is based on that handshake that we were talking about before. It needs to match, okay? So the first portion is kind of random. Like I said, it's based on the fact that the virus and the host cell kind of have to run into each other first. And understand that you as a human or any animal altogether are millions and millions and actually billions of cells, okay? So for that virus to finally find its ultimate host, it actually takes some time too. So just because it hits your face doesn't mean it's found its final host, okay? So for example, most of the respiratory viruses, like we're talking about right now, they need to find respiratory cells. So if it makes it on your toe, not really doing much there, okay? It needs to gain access to you. This is why, for example, when we're talking about these type of viruses, we're talking about mucosal surfaces. So eyes, nose, ears, mouth, that kind of stuff. And so this is where you hear the whole idea of, covering your mouth, and apparently people abusing things like masks for no reason more than anything else, right? So that's where that's really coming from. Now, for the bacteriophage that you're seeing over here, this is a virus that, again, infects bacteria only, right? Once it finds its host randomly, those little legs that you're seeing over there called tail fibers will actually interact with the host, will interact with the membrane and envelope of the bacterium, and then eventually it will actually inject its material, its genetic material, its DNA or possibly RNA into the actual host, right? So I wanted to kind of stop for a moment, and rather than giving you a story of coronaviruses, which I assume most people are kind of sick of this, I wanted to kind of just remind you of how many other big issues have occurred in the past. And in this case, one of the biggest issues that has happened have been influenza pandemics, right? And so even though what are we at, 100,000 cases or something like that we're worried about right now, these ones made it in the millions. And these are deaths, not just, oh, people that got infected. And the biggest culprit here happens to be this wonderful little guy that you're seeing here. And no doubt you've heard the terms H1N1 or some variation of those numbers. Now, remember when we were talking about the handshake thing, right? The handshake is about the viral host, in this case, virus and the host interacting, shaking hands in terms of their proteins. Well, in this case, these so-called flu viruses have two main proteins outside of them. One of them is called the hemagglutinin protein, which is the little pretty blue one that you're seeing in there. And then the other one is called the neuraminidase protein that you're seeing in pretty red, okay? Now, flu viruses that you're seeing over here happen to have both of these proteins. And so the way we usually classify them is based on that protein profile. So if they have version 1 of hemagglutinin, it's called H1. If it has version 2, H2, and version 3 is called H3, and so on and so forth. And the same thing applies to the N. Think about it in terms of humans in the same way. You're all humans, I assume. Some of you might be reptilians, you never know. But the way we kind of sort things out is based on how you display yourselves. And so whether you're tall, you're short, you know, that kind of thing, it's still a human. It's just how we categorize which proteins you're displaying. So hemagglutinin comes in several versions, and so does neuraminidase. So the very common one you've heard was this H1N1. And then you'll hear like H5N4 and so on and so forth, and then tons of variations. So now what happens? Every year, you're encouraged to take a, a flu vaccine, right? And everybody usually, and I'll let you guys do the same thing if you want to, usually asks, well, how come I have to get another vaccine? Why do I have to do it every year if I already got my vaccination? You know, it's still a flu virus. Well, technically, yes, it's a version of the flu virus, but those H1s and N1s or variations thereof all change every year. And depending on which profile does it have, it can be a little bit more infectious, a little bit less infectious, and so on and so forth. So what happens anytime you get a flu shot is that out of the several hundred that might be going around in the year, you might get the top 3 to 5 in that vaccine. So the profile from that H and N that you'll hear about, that's the shot that you're getting. And again, we are only giving you the top 3 to top 5 per year. The other 95 that might be floating around, you're not protected against those. So you still can get sick. But we usually give you the vaccine for the ones that are either the most deadly, the most problematic, or the most concerning of them all. Now, part 2 of this is the fact that, well, why do you do it next year too? Well, there's more, not just the 2, 3 that you got the vaccine for, but also that H and N are varying, are mutating as they go in and out of the host. And so when those change, your vaccine becomes kind of useless again. So you are going to get another infection by this flu type of virus or their cousins, including, funny enough, coronaviruses. So this is why there can never be a cure per se for a common cold. There's always going to be a cold going around. So despite the fact that people want to argue that you find a cure, there hasn't been one yet. Until we figure out a better way to address its genome as opposed to its proteins, we might find better solutions. So keep that in mind. Does that make sense, people? My little half story in there. Yes, no, I'm not seeing any faces hanging out with me. Thanks, cool. All right. So what this little virus, in case, again, don't memorize this, part of the flu viruses that we have is called the orthomyxoviridae, It's a negative sense RNA. It's single-stranded, for example, for this guy, and it comes in a bunch of chunks. So this is not a single sense. It has many little chromosomes, if you will. So this is what makes it a little more dangerous to deal with, right? The fact that, one, it's negative sense. It's not like us, right? It's using RNA instead, and it comes with multiple pieces. All right, so back to its getting inside. So it attaches itself, it recognizes and does the handshake piece, right? And then it needs to kind of breach, if you will, the wall or the membrane or both, right? So that means it has to poke a hole through it. And usually part of its capsid or its envelope, either one of them, helps it get through. Now, most of the time it possesses enzymes like lysozymes, like the ones we talked about during the cell lecture. And it starts kind of cutting through it until it makes a large enough hole for its genome to get into the actual host. Once it does that, then it overrides the system. Its genome kind of pretty much hijacks everything that is going inside the host. In other words, because it's injecting its own genome, its own RNA, its own DNA, it kind of starts stealing away from the host. So now the host, instead of doing its own job, it starts doing the job of the virus. And so what happens to the host, it stops working. It kind of shuts down, it pauses, if you will, while the virus is taking over all its pieces, okay? And so think about it this way. You're currently trapped at home, I assume, or in some enclosure, and then somebody else comes in the middle of the night and starts using all your stuff without you even knowing. That's pretty much what the virus is really doing, okay? It gets in, takes over all the pieces that you normally use, and it takes advantage of them and it starts making more. So somebody goes into your kitchen, makes its own food, and leaves. That's pretty much what it's doing. Now, again, there's a little bit of a difference between the ones that are bacterial versus eukaryotic. The ones that infect bacteria are usually called phages, in case you recall from a couple of slides back. And then normally the ones that infect eukaryotes and things like that are just referred to as viruses too. So this is just a convention, just so you know. They're still all viruses. Now, there is a little bit of variation though when they're trying to get into the host. So if it's a bacterium, for example, remember that bacteria have that cell wall. We don't have a cell wall. Don't forget that, right? So a bacteriophage, for example, something that infects bacteria, has to deal with it, whether it's gram-positive or gram-negative, that kind of thing. With us, not so much. But if they were to try and infect a plant, plants also have cell walls. So there's a challenge there. So there's a variation in terms of how it can infect its host. And one of the easiest ways to get in is whether or not that virus is naked or enveloped. When it's enveloped, keep this in mind, it looks like the host. Remember, wolf in sheep's clothing. So it pretends to look like it. And so it's more likely to be recognized and go, yeah, sure, come and hang out with us and not have a problem. So when the viruses are more or less naked, they kind of have to cut a hole through it so they can be internalized, or they can kind of be accepted through membrane transport pathways. If they're enveloped, on the other hand, they kind of fuse with the membrane and they just can be taken in. So the classic endocytosis pathways that you've seen before. So keep that in mind. All right, so just in pretty picture versions of this, you can actually see it, right? You're seeing a little polyhedral version on the top left-hand corner in which the virus just has to penetrate directly into the host to inject its content. Whereas on the right-hand side, because it has that sheep's clothing that I was talking about before, it can fuse with it, be identified as if it was part of the team or the family, and then be taken in. So keep that in mind. All right. So now it gets in. Now it has to kind of hijack the system. It has to use its blueprint, and its blueprint has to take advantage of all the host's materials, all its host machinery. And so one of the things that happens is that, for example, the polymerases, your DNA polymerase, your RNA polymerase, or whatever it is, get hijacked to make the virus's material. Sometimes the viruses can come in with their own, just so you know. And so what will then happen is once it produces all the pieces, all the proteins that it needs to do so, then it needs to assemble. And so what you're seeing down there is just kind of a little bit of an assembly of what it will look like for a bacteriophage, a virus for a bacterium. Now, usually this is one of those times that somebody comes in and asks well, how does it do that? And the answer to that is we don't know. This is one of those kind of levels of material that we're still kind of looking into. Remember, these happen insanely fast, so timing happens to be one of those challenges too. So, and then separate from the fact that, you know, nobody wants to volunteer to be infected by a virus to see how it happens, right? All right, so let's go back into a little bit of the central dogma and talking about gaining access, going through the central dogma, DNA, RNA, protein, that kind of thing. Here's what we really need to deal with. So the issue here is that once it gets in, it needs to make more of its genome, so replicate its RNA or DNA, negative sense or positive sense, it doesn't really matter, okay? And it also needs to kind of deal with whatever is inside the cell. So remember, eukaryotic cells have tons of organelles. Most bacterial ones are missing most of those, right? So depending on what's inside, it will kind of deal with that type of adaptation of the host. So kind of keeping this in mind that, for example, eukaryotic cells have a nucleus. And so they need to be able to access where that material is. Whereas your prokaryotes, their DNA is usually kind of free floating. Remember that their DNA is circular as opposed to ours as being linear, that kind of thing. All of these pose challenges for how the virus can take over and hijack and get out. So reminding you that there's versions of the senses. The idea here is if it's positive sense, it can kind of go directly into making protein. If it's negative sense, it has to go back and make its complementary strand and make more. And remember, whether it's DNA or RNA, those all kind of vary. So those are the variations that you're seeing up there. Okay. So now, it hijacked, it assembled, now it needs to get out. So this is your exit, your release. And pretty much this is the worst part of this, is that the cell gets ultimately destroyed. So usually by using some of those lysozymes that we were talking about before, those enzymes that destroy the walls, one of the biggest issues here is that since it makes hundreds, if not thousands of itself, it kind of starts burning the cell itself until it kind of bursts. And at that point in time, it releases everything out into the world. Now, here's a little bit of an issue here, is that sometimes it can just get in, hijack, get out, and can do this in a matter of minutes. The time that it does it from attachment to getting out is called a burst time. Now, sometimes though, it can actually stay inside and not explode the cell. So that's going to be the next step that we cover. Okay, but before we go on to that, just briefly remember that time that it takes from getting in into getting out, sometimes just a matter of minutes. This is referred to burst time. And how many of those kind of manage to get out is usually the burst quantity that you're seeing on the lower left-hand side is how many of those can actually, how many of those viruses can be released. So what you're seeing here is just kind of like when we looked at bacterial growth, here we're looking at viral reproduction. And so in a matter of just a couple of minutes, and notice that that little scale over there you're seeing is 20 minutes. It's the time of our burst time from getting in to making thousands of itself and then getting out. That's what we're talking about. And how many it releases is referred to as its burst size or burst quantity, depending on which one you want to use. All right, so what happens when it stays inside instead of blowing things up? So there's a modification in which normally that kind of getting in and getting out of 20 minutes or so is referred to as a lytic cycle because it's just blowing the cell up, so lysis, right? But there's a version called lysogeny in which rather than getting in and getting out really fast, it takes its sweet little time inside. And here's where it gets dangerous, and here's the part 2 of what we talked about in the first portion of this topic when we were talking about cancer. Sometimes, rather than getting in and getting out, it takes a nap. But it takes a nap by taking its genome, its DNA, its RNA, or whatever it is, and it integrates itself. It becomes part of the host's genome, okay? In other words, it becomes part of the host's DNA. Now, Why would that matter? Why is that a big deal? Well, first of all, if it managed to incorporate itself inside the host, it's hiding again. So even, for example, the host defenses wouldn't be able to find it because the cell is not technically exploding or doing anything bad. It kind of goes a little silent. It's just chilling inside the host itself, hiding, plain sight, basically. Now, what happens though is the second advantage is that because it became part of the host's DNA or genome, anytime that host replicates, in other words, it doubles itself, it actually replicates the virus too. It does its job for it. In other words, once the virus becomes part of the host, that host, as it divides and makes more copies of itself, it's making also more copies of the virus for free. And that's a really cool concept. Now, what happens here is that sometimes the virus can become part of it permanently and never get out. And what's interesting about that is that that makes that host a little bit more dangerous. For example, in bacteria, if some of the proteins that the virus makes are toxic for some reason, you just created a stronger bacterium instead. But what's kind of gnarly about this lysogenic cycle, which is what you're seeing on the right-hand side, is that it'll just hide inside the host until it finds the right time. Now, to define the right time is a little bit more iffier. But the idea here is that it incorporates itself and it can hide inside the host for years. HIV happens to be one of those, for example, in which can hide inside a host for years without causing any damage, any issues, and then it chooses just the right time to make more copies of itself. And here's where it gets really, really ugly. So I'm going to kind of switch off to a little bit of a side screen. Let's see if I can do my, looking for my screens of death over here. Let's go with a black screen of death. So imagine one little virus over here, and I'm going to put a second little virus over there. Okay. And so normally when this virus gets inside a host, it will make, you know, a few thousand of its own little children, little progeny. Okay. But in the alternative version to this, its little virus can integrate itself in part of the host. It becomes part of it. Let's see if we can show this a little bit better. And incorporates its piece into the DNA, into the host itself. So that's part of the virus. So we call this lysogenic. Whoops, I just lost my slider. You guys still there? It kind of glitched on me really quick.
Yeah.
All right. Not sure why it happened, but we'll get back to it. Let me find my awesome little drawing that I was having way too much fun with. You guys still see my awesome little drawing, right? Yes. On the screen? No?
Yeah. Yeah.
Yes.
Okay.
Yeah.
People are saying no, like, wait, what's going on? All right.
Yeah.
So now, this is called lysogeny. There it goes, lysogeny. And so what happens? This cell, this host, let me see if we can find it. I'm gonna make up a new cell over here. This cell that has its own leader DNA kind of contaminated, if you will, with some viral DNA, will just replicate on its own. Not worrying about the world, not damaged, not hurt. And after a few hundred divisions, remember that every single one of those cells has the virus in it. And just at the right time, then each one of those cells can make a couple of thousand of viruses at the same time. And so when this guy explodes, it really kills everything. It takes down everybody hard, fast. Because not only are you just making a burst size and a burst time of 20 minutes for 1,000 viruses, here you're getting millions and millions and millions all in one shot, one quick hit. So that's what makes it far more dangerous. All right, so there's two versions. Again, the straight kind of quick and fast version of it, called lytic, or this other version called lysogenic or lysogeny, okay, in which it kind of hides inside the host, waits for the right time, and then explodes into tons and tons of them a little bit later. All right, so now, how does this look like in terms of our viruses? So remember, we were talking about examples in terms of the bacterium up there. Eukaryotic viruses, remember that for us, we don't have a cell wall in terms of humans, although plants do and things like that too, which means that, for example, our viruses are a little bit more prone to having those envelopes simply because they can steal it from us a little bit more often. And there's really not a lot of variation in terms of our genetic material versus theirs. So their strategy is still the same, still like it is in terms of bacterial viruses, but they have a couple of advantages. They can kind of get in a little bit faster. So the same concept, there is a lytic cycle behind that, so that doesn't change, but they can have their version of lysogeny, but in eukaryotes, this is called latency. Same thing, just a different name. So in a bacterium, we call that a lysogenic cycle. In humans, for example, in plants and other things, that's called a latent cycle. So again, going back to something like HIV, it's a latency that we worry about, okay? And so when it integrates itself into its genome, when it goes into the host and becomes part of it, when it's a bacterial one, it's usually called a prophage. When it's a eukaryotic one, it's usually called a provirus. Again, they mean the same thing, okay? It's just fancy little terms that we use to differentiate between the two. Now, here's the dangerous part. This is really where we've been kind of leading all along, is what happens when it integrates itself, what happens when it goes latent, but it never comes out, meaning it never bursts and never leaves the host genome, meaning that there's no induction of that virus to take over. In other words, it becomes part of you. It becomes incorporated into your regular genome. So that's where it really gets to be fun. And so we enter this kind of wonderful little field of cancer. All right, just a little side. For those of you guys that want to talk a little bit more about cancer, we can talk about that later with more AMP side behind that. But for those of you who recall, cancer, which happens to be an unregulated or dysregulated version of cell growth, meaning that your cells start growing uncontrolled, if you will, has different origins. There's different reasons typically associated on why the cell can do that. Either you're feeding it too much, either it's, loses its stops, things of that sort. But all of these have the same concept. They lose their ability to stop growing, to stop dividing. And so viruses can do the same thing. And this goes back to our example that we gave you with HPV, right? Some of these can trigger cancer. Why, how, where, when? This is kind of where we get to have a little bit of fun, is that remember that during transcription and translation, everything has a start and stop codon, everything has a promoter and a terminator, and so on and so forth. Well, remember that in order for you to begin transcription, you need a promoter. You need a place to start. If there's no promoter, there's no place to start transcription, okay? So what happens? What if you mess with that promoter? What if the virus, when it goes latent, when it gets inserted into our genome, messes with those controls? In other words, it changes things on how they grow. And so I'm going to try and use another screen. That way I can demonstrate this a little bit better. I'm going to use a white screen in this case. Okay. Let me try and remember that everybody has your, us as a host, nice little chunk of double-stranded DNA. And remember, there's a portion of it called a promoter, and there's a portion of it called a terminator, where to start and stop transcription. In other words, your RNA polymerase, I'll make a little protein over here. Your RNA polymerase knows to start over here and it knows to stop over there. And if it does that correctly, it will make a little chunk of RNA. Okay? And then eventually, that little chunk of RNA would also turn into a little protein. We've been talking about this before. Now, what happens if, I'm going to use our DNA of our virus in purple. Whoops, went too far. And I was having fun drawing. What happens if your viral DNA, I'm going to just kind of, let's call this a virus DNA, happens to insert itself into that sequence that we just had earlier. And let me give you some options. Here, I'm going to put this chunk over here. Here's our promoter. I'm going to put my terminator over here. Oops, that's supposed to be a T. Sorry. What if it inserts itself somewhere in the middle of that? What if the virus chunk of DNA goes in here? Okay, here's an interesting issue behind that. Is that if it happens to do so, okay, I'm trying to check I saw the chat, but I lost it over here. That's my little chat. So anyway, if it gets in the middle of that, that means that the RNA that it's going to produce, trying to get this guy, the RNA that you're going to make is going to be pretty much disrupted somewhere in the middle of that. You're not going to get the right protein. You're going to just build something weird and more likely just bad for the cell. But every now and then, it can actually insert itself right where the promoter is. The one that decides if it starts or not. Now, that may not be seem too bad, but what if that protein that we're having to make, this protein right here, whatever this may be, happens to be a protein that is responsible for cell growth or cell division. Skip an I over there. There it goes. Now that virus has literally interrupted the sequence. And when something should be growing or not growing, this virus is messing with that control. And so this could cause the cell to now start overly dividing, start growing and growing and growing and dividing uncontrolled. So what is it that's the issue here with HPV? HPV happens to infect a particular tissue that divides a lot, the cervix. That tissue regenerates itself constantly. Remember that whole, monthly thing that most of you should be familiar with by now? So that type of cell is generating lots of tissue quite often. And when HPV happens to be present, it gets more chances to incorporate itself into those type of promoters. And when it does, now it can cause that tissue to grow uncontrollably, leading to what most of you know as cancer. Now, if it did it on your skin, you get a little wart. It's an uncontrolled growth of skin. But if this manages to make itself into uterine tissue, that's a bigger deal. That's really where a much bigger portion or picture in terms of disease comes into play. And going back to alluding again to sexually transmitted infections and things like that. So this is a big deal, people. All right. So let's leave the last little pieces that we have for the next few minutes to how do we grow it. One of the bigger challenges is, as opposed to bacteria in the lab, which we just get some agar and throw it in there, pretty easy. But remember that parasites need hosts. And since none of you seem to be volunteering to be infected by viruses, that makes it a little bit more challenging. So we usually use live organisms as a way to host them. In the case of bacteriophages, well, we just make some E. coli and we can grow them on those. Nobody seems to be crying for the rights of E. coli. But when we want to use bigger things, we can use plants. That doesn't seem to be anybody protecting plants that harshly these days. But what if we need an animal? Well, we found out that one of the best ways to grow eukaryotic viral parasites in this case are eggs. you can actually take eggs that have been embryonated, meaning these are not your classic eggs you buy at the store. Those haven't been fertilized. These ones have been fertilized and are starting to divide, okay? And you can actually introduce the virus, depending on which type of virus you want to grow, into certain areas of the egg, and the egg will grow the virus for you. You can have millions of eggs, actually, in little racks, just like it looks like the store, but each one of those fertilized eggs can be actually growing viruses for us to study. The other version of this is we can use primary cell lines, so tissues that we've obtained from eukaryotic hosts. Some of those can be human. These are called primary cell cultures. Most of them are usually kind of immortalized, so we can grow them and grow and grow them. And so we can use those in the lab too. So on the left-hand side, you're seeing kind of what happens when you have like a little lawn of E. coli being infected by a bacteriophage. You see those little plaques, those little holes telling us that it infected and killed those bacteria. And on the right-hand side, you're kind of seeing which versions of a fertilized egg you can actually do that to host eukaryotic viruses. And then ultimately, like I said, we can have immortalized cell lines that we've taken from some sort of eukaryotic host, and those cells can be used as a way to grow viruses too. All right, so that's kind of what we're shutting down all the material that we really wanted to talk about classification in terms of viruses. And again, if you guys want to hear a little bit more about the whole wonderful little world of COVID-19 kind of stuff, we'll talk about that a little bit later. But now, viruses happen to be one of those groups of tiny little things that, you know, are parasitic and destroy us, but there's more. Believe it or not, there's a group that follows very similar patterns to viruses called viroids. And in this case, the crazier thing is that they don't have a container at all. So remember, your viruses need that protein coat, that capsid, right? Made up of those tiny little pieces called capsomeres. Viroids, none at all. They're flat out naked, okay? This is a little chunk of typically RNA, okay? No capsid, no coat. They're typically circular pieces of RNA. They're much smaller even than your classic virus, so that's very tiny. And they're very much infectious too. Now, interestingly enough, or at least fortunately enough, we typically have only found them in plants. We haven't found these in humans yet, okay? And then there's a similar version that occurs for fungi, in this case called viroid-like agents. So those kind of occur there too. But really, the one that I wanted to talk about at least briefly, since we will talk about diseases later, and you guys will have a project for that, considering we don't have a lab, that can, you know, test these, are the group called prions. And this is one of my favorite ones I usually talk about, especially when I took virology and I took microbiology. Even my instructor was a little crazy and told us the stories on, you know, in the late 70s, early 80s, and into the 90s, in which people tried to demonstrate that these existed. And no joke, everybody just laughed at them. They were ridiculized, they were kind of barred, until we kind of found out that they were right and they won the Nobel Prize. So the person responsible for studying prions, his name is Stanley Prusner, in case you're interested. And what he discovered in his group, in his research, was that there were proteins that could be infectious. Not a virus, not a viroid, not a cell. It was just a protein. I want you to understand how crazy this is. There were no DNA. There was no RNA. None of it. It did not require a genome. It was just a protein that can mess with another protein. Okay? Now, remember that all proteins function by way of their three-dimensional shape. Remember this whole denaturation thing we're talking about. Now, these proteins purposefully denature other proteins. Now, I shouldn't say quite purposefully because it sounds like they have a mind of their own, but their actual shape messes with other things' shapes. Now, what's the big deal behind this? I assume that at some point in time in your life, you've been told this. I've been told this all my life, that, you know, depending on who you hang out with, we can tell, you know, who you are, kind of thing, all right? And they all will always tell you that, you know, you shouldn't be hanging around with the wrong crew, kind of bit. Well, this is this guy. This is the one that corrupts you. This is the friend that you shouldn't be hanging out with, but you still hang out with anyway. Prions are misshapen proteins, are technically denatured, right? But cause other proteins to denature themselves too. They corrupt them, if you wish, okay? And so what will happen is that if you have this bad protein hanging out, it gets close to the good protein, the one that's not doing anything bad, and it'll reshape it and make it do bad things too. Again, no joke, it's corrupting it. And what's crazy about these proteins is that they're also heat resistant. So remember that heat usually denatures things. Heat typically does not change the shape of these guys. So why is this the biggest deal? Well, we talked about this on day one of this class when we were having a little bit of fun talking about what type of things we're going to be covering. And Sam, you just kind of threw that in there, is yes. For those of you who heard of something called mad cow disease, and we talked about our transmissible spongiform encephalopathies, or TSEs, right? These are it. You see, things like mad cow disease are not a virus. It's not a cell messing with you. It's your own corrupt proteins corrupting other ones, okay? So what is actually happening here is that one protein kind of goes bad and it takes the rest with them. So what happens with the whole mad cow disease concept? And allow me to kind of show you this in the last set of slides that I have over here, is that these proteins, these prions, are already damaged. They don't do their job. Remember, all proteins are about function, or at least structure. So what happens when these ones are misshapen, they start kind of clustering around each other and start leaving holes. Now, what happens when this makes it to your brain? Well, it starts kind of turning your brain into cheese. What ends up happening is these proteins start clustering themselves, corrupting themselves in this kind of little domino effect, and ultimately poke holes into your brain, causing you to go a little crazy. Hence the term mad cow disease. Now, we've known about these diseases for quite some time. Probably one of the most famous of them all is called scrapie, which was found in sheep. And so a group of sheep happened to be kind of doing something really weird in which they would jump against fences and walls and typically barbed wire and scrape themselves, usually harming themselves and sometimes killing themselves. And so what happens is that, well, these are kind of weird sheep. So usually the owners would say, okay, we'll kill them, grind them up, and feed them to the next generation of sheep. Well, it just so happens that, again, grinding them up does not really destroy the protein, and nor does cooking it. So when these other sheep would eat it, they also went crazy. So they did that, they passed it on to cows, cows went crazy, and so on and so forth. Humans ate the cows, and you see where we're going with this. Now, what's interesting about these is that there seems to be a genetic predisposition, meaning that certain people are more predisposed to having that type of corrupt protein, misfolded protein. And we've known about this because there's groups out there in the world that actually have this happen to them more often. Probably the most infamous of them all is called Kuru, in which certain Aboriginal tribes, again, they stay away from everybody else. One of the things they thought was really cool, I think it's kind of cool too, is that, you know, when they would murder another little tribe and say, we killed you, we conquered you, they would cook them and eat them, right? That whole idea of absorbing their power concept. And guess what? Favorite part to eat? Brain. And guess where that protein happened to be? Brain. So this kind of actually started happening quite more often. So in any case, small little versions of exceptions to the central dogma again, and that are still infectious. They can still transmit themselves and still cause you a really, really bad day, but typically a little bit longer drawn. All right, so we'll stop right here since we have all the material that I really wanted to cover. That is the last little bit of virus stuff. There's no more in the lectures. And as we speak, like I said, I'm still trying to write stuff up. And what I'm now doing for the recordings is I'm writing a transcript. Remember that YouTube doesn't usually close caption unless it can detect what you're talking about. And you see me going crazy over here. So I'm writing a separate set of notes that I'm typing out for you guys to have too. So in case you can't hear me or whatever reason, you'll also have that transcript as well. Now, the plan is exam, right? Remember that the two review sessions, I'm sorry, are tomorrow, okay? So all I need you guys to do is, if you want to join, send me a quick email so I know to expect you. I probably will change the password to those rooms on Zoom, just so you know. Again, trying to prevent any terrorist attacks. Make sure you participate to one or both. The review guides themselves have been up, so that hasn't changed. And just remember that for everybody, especially your group, should be meeting on Wednesday on Canvas. Remember that, to make sure you take your exam from 11 to 12:15. That's your window for taking your exam. And it will be password protected. And so all that material will typically come out tomorrow in announcements. So make sure you check those announcements, okay? So before I move on to the next lab, which are my other people, I'd say just hang in there, don't go away. But let's make sure I kind of open up the room for a little bit of a conversation, since I know some of you guys might have some questions still going on. So talk to me. Stop the recording.